Good to see you as well, Chair Benson, and thank you, committee members, for inviting me here today. My name is Ping Ye, and I'm the co-founder and CEO of StemoniX. And before I begin with the 2 to 3 slides that I have prepared for you today, I'd like to share the backstory on the creation of StemoniX. And it was really birthed through my experience as a cancer survivor. So 7.5 years ago, I was diagnosed with Hodgkin's lymphoma. And the first 8 rounds of chemotherapy did not work for me, and had to be given a more potent version of chemotherapy that was very toxic and more toxic than my previous regimen, and I nearly died from that treatment. And so when I was laying there in treatment, I often thought to myself one critical question and one critical problem, which was, how can we test medicines on human cells without risking the lives of the patient? And from that point on, StemoniX was born. Yeah. Oop. There we go. So this is StemoniX Maple Grove. It does not look like this today on an early March day, but StemoniX, if you ask the industry how they see us, it's really as a company that has converged biology and software. It's a company that it makes human organoids, and I'll talk a little bit about that, which is the ability to convert skin or blood into a form of stem cells. That won the Nobel Prize in 2012, and I was able to license that technology after my experience as a cancer survivor. I'm a nanotechnologist by training, and so I wanted to see if my background could be useful in the life sciences industry, and that's how the company was born. And I formed a team that really were on the forefront of stem cell technologies, and the main challenge was, how can you industrialize these processes so that you could help the drug discovery industry test compounds that could potentially work for all of us. Currently, millions of animals are sacrificed a year to test if these medicines will work on people, but they are often not the right models, especially for neurological diseases. So StemoniX converges biology and software, and here's one of our products. This is a plate, international standard plate. It's filled with organoids of different formats, brains, hearts, pancreases, and they come in different formats. And once again, these do come from skin, so there's no embryos that are used to make this process. Now, we generate so much information from these organoids, and you'll see in a few seconds here what I mean by that. We need a lot of software to analyze that data. And so we're really seen as the leader in the world now in industrialized human organoids, and it's really maybe best exemplified by this picture. Every dot that you see here is a human cortical mini brain, as they say in mainstream media. Each dot is about 40,000 brain cells, and we brought on really the leaders in the field back even 10 years ago. The cover article of Cell was led by one of my lead scientists. And what was interesting here is once we actually made industrialized versions of these mini brains, we discovered a couple years ago that we could form, if this video plays here, you'll see a flash of orange light there. That essentially is an early form of brain wave in these mini brains. And we used these brain waves to test drugs, whether for safety or for discovery. We have the same for heart as well. You can see one of our mini hearts here looks just like the plate on the left, but they beat instead of communicate like the mini brains do in that video. To give you a sense of what can be done with this, if we plotted out this pulse at the top, you'll see this sinusoidal pulse that you can use for testing drugs like safety. So if that signal that you see at the top there deviates from normal, you know you've got an issue. And we're delving into ways of using that to determine what types of actual mechanisms of toxicity that are causing the issue with that drug. Now, what's really interesting with this technology is that this waveform that you see on the bottom is from a child with a neurodevelopmental disease. So what we're learning is that children or people with particular diseases and the brains that we make from them can actually have a fingerprint of their own waveform, thus giving us an opportunity to test thousands of compounds to figure out what of those compounds changes the biology to ameliorate the disease. And so here you can see at high scale, nearly 400 brains on a plate. You're seeing hours of data across all 400 of these brains. It's like an hour-and-a-half movie, if you will, in a single 400 hour-and-a-half movies in a single plate. And so we have to apply a lot of software, else it would take months by scientists to analyze this data. And essentially what we've done is shrunken the hundreds of hours down to 2 minutes using augmented intelligence, AI approaches, and data analytics. So that's really our advantage out in the marketplace, is this ability to make these organoids that are highly functional, that can screen thousands of compounds for the whole industry. Funding-wise, I'm very proud to be here because Minnesota, the state of Minnesota, has been a huge part in the history of StemoniX. We were honored with the recognition of winning the 2016 Minnesota Cup and have been participated in the Angel Tax Credit Program. I think we were the top recipient back in 2016, and so it was a huge part of getting us to where we are today. And with regards to where we are today, I can say that we're making efforts to help even on a national level. So I'll skip over the fact that we use AI to find drugs and all that. I don't know if we have time for that, but I can say that we were chosen by the National Institutes of Health as the brain model of choice to find painkillers that are less addictive in the national effort called HEAL, which is helping to end addiction long term. So they're using our brains to find painkillers that are less addictive. With regards to our traction, stemming here from Minnesota, we now serve 15 of the top 25 pharma in the world, serve government agencies, including the U.S. Army on fighting weaponizable viruses like Ebola. Our total paying clients now is 80 globally. And essentially, our products and services are helping our customers and partners save hundreds of millions, if not billions of dollars, which really gives us and them potentially the opportunity to address drug prices by giving them the opportunity to more efficiently get drugs to market. And very happy to say that 2 top 10 pharma companies will be presenting and submitting papers in peer-reviewed journals in the coming weeks. Here, a snapshot of some of our customers. Half of these we work with on a weekly basis. And I think just to tie up the ends here, we have been able to still be on the forefront of the leading journals. This was the October issue of Cell Stem Cell. One of my leading scientists was able to make a more complex version of brains that you see on the left there that has a subcortex. And essentially, this is also made from skin, but the electrical activity of this organoid had the similar to an EEG signal of a 35-week-old premature infant. So these are ways we can test drugs without actually risking the lives of people and children. Essentially, if I were to summarize everything that I've said today, StemoniX is essentially at the convergence of stem cell technologies, human organoids in one area, software analytics and AI in another, and the ability to screen thousands of compounds, including the ability to program or gene edit these cell lines. And so in summary, together we believe we can unlock some of the most challenging diseases that face society today. And here's a snapshot of the team. I'm not much without them, and it's an honor to be here with you today. Thank you very much.
Thank you, Mr. Ye. Senator Kiffmeyer.
Thank you, Madam Chair. This is an amazing hearing to be able to know about some of these phenomenal things. So thank you very much for being here and telling us about StemoniX, rather. So I have a couple things. One of the things that I take a look at, especially in your brain thing demonstration material here, and that is, have you done any work on seizures and vaccines?
Chair. Oh.
Mr. Ye?
Yeah, Chair, Senator. We have. Actually, seizures was one of our key core competencies, and we use our brain waves to test drugs if they cause seizures, which is a major issue. And for those that aren't familiar or live and breathe the life sciences industry, the #1 cause of safety failures in clinical trials is actually neurological safety issues. And so there was a paper in Nature a few months ago that showed that mouse brains just are not the same as human brains. It might not be a surprise to many of you, but it
Thankfully
is
so.
genetically. And so this ability to make human-like species-specific models can really answer the questions around seizures and the types of diseases that we're going after. Right now, if I list them out, autism spectrum disorder, Rett syndrome, which is a form of seizure, schizophrenia, various forms of dementia. We just signed a couple contracts in the area of dementia, Huntington's disease, Alzheimer's is something that we see coming forward, and we actually have the ability to make Parkinson's brains as well. So this is truly a platform. With regards to vaccines, we worked with US AMRID, that is the US Army Infectious Institute for Diseases. They look at weaponizable viruses, and they've used our brain cells actually to infect our brain cells with Ebola, dengue, Zika virus, EEE. We just reached out to them a few days ago to see if we could be helpful in looking at the toxicity profiles of potential drugs to treat coronavirus. And you can use our models the other way around, not just as an infection approach, but also find vaccines by making using our model as an inhibitory model. So giving the vaccine first to our cells and then introducing the viruses and seeing if it can inhibit infection. So yes.
So, Madam Chair, thank you very much. That was an interesting follow-up. Seemed logical when I see the human brain. But this is more, I would gather, preclinical. You still need human actual trials, but it kind of expedites that initial thing to narrow the sorting out process to drugs that are valid, so you don't have this super long time and then find out it's not really working?
Absolutely. Chair,
Ye.
Senator, you're absolutely right. This is really about compressing preclinical time. It can take years in animal testing before you actually get to human trials. If you look at phase 2 statistics, 55% of the time in phase 2, drugs fail due to not being effective. And 30% of the time in phase 2, it is unsafe. So you're dealing with a massive failure rate in phase 2, and by that time, the company has already saved or spent hundreds of millions of dollars to even get to that point. So the ability to, if you will, create a clinical trial on a plate of phase 1 or phase 2 would basically be a paradigm shift and a transformation to the whole industry.
Thank you, Madam Chair.
Senator Kiffmeyer.
I would say, considering that you did a lot of this thinking while you were very sick, goes to show you had some good brain activity yourself. And we are grateful for that. And sometimes when you're flat on your back and you have to, just thinking of some of the good that has come out of that. So commend you for being so productive even while you were so very sick. Thank you.
Further questions? I want to thank you. Could you, on the slide you showed the plate, could you show us how big that plate actually is?
Sure, Chair. It's about a little bit larger than your large iPhones or phones that you have in your pockets. So it's an international standard plate, and we fit about 400 brains or hearts or pancreases in each one.
And Mr. Ye, you had shown me, I believe, a slide where you grew a tumor.
Yes.
A brain tumor, so that that could be used for testing effectiveness of chemotherapy or gene therapy.
Yes, Madam Chair. We had, a lot of us are touched by cancer, not just me, but parents have passed away from cancer. So one of the questions we had is, could you introduce tumor cells into these wells or little petri dishes on a plate to basically mimic metastatic active cancer attacking healthy cells? And we were able to do that in a concept. We took glioblastoma, brain cancer, and introduced those tumor cells into the same well as a single mini brain. And we figured out a way to keep both alive long enough for the tumor cells to infiltrate and attack the healthy brains. And the corollary to this in the animal world is you have to humanize a mouse, you have to inject tumor cells on the back and see it grow. And I think there are ethical issues with that, but instead of killing thousands of mice in a single study, you can take some skin a few months later, have a plate of brain tumor cells that you can screen thousands of compounds. And I'll say that the question that a lot of people ask next is, well, is there a personal precision medicine approach with such technology? And really, there's nothing in the way of us doing that technologically.
Thank you, Mr. Ye, for your time.